First, hello and welcome to Chasing Electrons, dissection of early cold-induced deficiency in soybean. During this webinar, we ask that you keep your microphone on mute for the duration of the presentation and put any questions related to this presentation in the chat so they can be addressed accordingly. There'll be ample time to address questions after Dr. Robison's presentation. For any questions we're unable to address during the webinar, we'll be sure to follow up directly with you via email. Please allow me to introduce Dr. Jennifer Robison. Dr. Robison is an assistant professor of biology at Manchester University. She's been teaching undergraduates for over a decade, and her research specializes in the physiological and molecular response of plants to abiotic stress. Outside the lab, she's a single mom and an amateur glassblower. So I'm going to let you take it from here, Jen, and the floor is yours.
Thank you, Carrie. So I'm really excited to talk to everybody today. I am going to be telling you some of my story, both professionally and personally, and then the Handy PEA I have been using. I've kind of been a big fan of the Handy PEA since 2004 when I was a master's student, and it was one of the pieces of equipment I had at the top of my list for my startup funds. So as soon as I got my permanent position, I started begging for them to buy me one. So I'm going to tell you how I use it and why I think it is so incredibly cool today. If I can get my screen to, there we go. So some of you are probably wondering, where is Manchester University? What is Manchester University? It is a very small liberal arts college in the Midwest of the United States. So we're located in Northeast Indiana, about 2 hours northeast of Indianapolis. We have 1,200 undergraduate students, and that's it. So my lab for the last 4 years has been using undergraduates exclusive. I have 2 to 3 research students a year, and this is my group so far of my 4 years. You can see all those wonderful faces, and the little boy in the red shirt is actually my son, who comes in and trains the students how to plant soybean, because he's been with me since day one of my PhD. He was a first grader and came into the lab all the time. So he's my resident expert on soybean care and maintenance, and he comes in and makes him feel very special to come in and train all these wonderful students. As Carrie said, my lab focuses on abiotic stress. For today, we're going to focus on cold stress in soybean. I also have a mechanical wounding project with soybean that we've just started, and I'm interested in cold germination in an invasive species around Manchester. Other than myself, there are a couple other people that I want you to know their names. So Ali Schumacher, she graduated from Manchester in 2020. She's currently a PhD student at Michigan State University, and she was my first student to work on the photosynthesis project. So you're going to be seeing some of the data that she collected. And my old PI from IUPUI, where I got my PhD, Dr. Stephen Randall, we did part of my PhD was the beginning of looking at some of this photosynthetic information. So he was very helpful and influential in getting this data that you're going to be seeing as well. So like I said, cold stress, soybean. Why soybean? Soybean, if you don't know, is an incredibly versatile and valuable crop species. So I took this picture at the Indiana State Fair because it really sums up soybean, right? It can be used for food. If you eat tofu, it's used for animal feed, but it also is used to make crayons. It's made sunscreen. You can do paint, biofuel. The elevator oil that runs the Statue of Liberty elevator is made entirely out of soybeans. It can be used for ink. It can be used to make clothes, right? So it's a lot more than just tofu, right? Soybeans a lot more, gives us a lot more than just that. And when you look at the United States, soybean is the 4th top commodity. And it's the 2nd when we're talking about crop species. So it's worth $40.3 billion in a year, as according to the USDA in 2017. So soybean has a limited cold tolerance. Unlike other species, it's unable to undergo enough physiological and molecular changes to acclimate and survive in the cold. Some of you probably are familiar with Arabidopsis. It's the model plant species, and it is very cold tolerant. So it's chilling at -5, -6, and it's like, yeah, this is awesome, right? Soybean, it hits 10 degrees and they start withering and getting really, really not happy. So this is kind of my little image of Arabidopsis being happy in the cold and soybean being sad in the cold. And when I say that they don't do very well, this is 3 pictures at 4 degrees. So Arabidopsis is fine at freezing. This is 4 degrees. And even after 24 hours, the leaves are starting to droop. You can see they're getting paler. By 2 weeks, you have full-blown tissue damage and the plants are dying. And when I looked at the actual lethal time for 50%, it's 5.2 plus or minus 0.6 days. So we actually did this as part of my PhD. I have a paper that shows how the different types of soybean, and pretty much they all have about the same LT50. So after about 5 days in the cold, soybean are dying, which is not good for crops and agriculture. Now, we know that cold temperatures disrupt photosynthesis, and so I just wanted to remind everybody and familiarize everybody with the Z-scheme in photosynthesis, because it's going to be really important to the measurements that we're going to be talking about in a little bit. So we have, right, photosystem 2 gets electron, gets photon, excites that electron, it pops up, and then that electron gets passed down to photosystem 1, and then more light makes that electron get more excited again, and then it eventually gets passed down to fix NADPH, which goes to the Calvin cycle. In the cold, you have electron transport rates decrease. So that movement of the electron between photosystem 2 and photosystem 1 starts slowing down. The PS2 reaction centers become closed, and so you have less PS2 reaction centers that are capable of sending those electrons. And the reaction center repair is slowed down. So you end up with more broken PS2 reaction centers that can't be fixed. Overall, PS1 efficiency decreases as well. So really in the cold, we're impacting almost every part of the Z-scheme. And that's known for most plants, right? Like depends on what their cold stress is, right? This happens in Arabidopsis. It's just at a lot colder temperature than it's going to happen in soybean. We're going to be using chlorophyll A fluorescence. Well, that's what the Handy PEA does. And so a real quick basic in case you are new or, you just kind of forgot, because I do that all the time. It's like put something away for a while and forget about it. When light gets captured, it goes 3 places. It either goes into photosynthesis, it's non-photochemically quenched, usually dissipation as heat, right? So it leaves the system, or that light bounces back as fluorescence. And it's that bounce back that the Handy PEA and your other fluorometers are actually detecting, right? So it's that bounce back fluorescence that you can see, and that's why we can then calculate how much energy is being used in photosynthesis, how much energy is being dissipated away from photosynthesis. When you look at just a basic fluorometer curve, right? So this is just one I drew a long time ago for a class I was teaching. I just drew it on paper because that's how my technology was at that point, a long time ago. At the very bottom at FO, all the PS2 reaction centers are open. They're ready for electrons. And then you hit them with a burst of light and all the reaction centers become closed, and that's FM up at the top. And then we can calculate that maximum efficiency by looking at the difference between where it started when they were all open and where it ended when they were all closed, compared to that peak, that FM. So that's FV over FM, which is the maximum efficiency. Now, this is all taking place with a dark adapted sample. So you put the plant or the algae or whatever in the dark for a little while, let all those reaction centers relax and open back up, and then you hit them with a bright light. You can also do this in the light, and it's usually in the literature, it's going to have like this little apostrophe that lets you know, oh, this was done with the light. So if you look towards the right of the screen on my little graph, you can see the light is on. Those little orange starbursts are supposed to be light. And so you would look at the FO would actually be that really low one, and the FM is the jump, right? So that's in the light. The Handy PEA is what I use to measure chlorophyll fluorescence. The cool thing about this is it captures not just that FO and the FM point, but it captures the kinetics all the way through from FO to FM, right? So it gives you a lot more information than just here's the overall efficiency. And when I say it gives you a lot more information, I mean it gives you a lot more information, right? So this is just a fraction of an output from the Handy PEA, and my undergrads look at this and go, it's like, okay, it's fine. Don't worry. I don't expect you to understand all of this. We're going to go through it and I'm going to help you figure out what the important parts are. So it gives you a lot of data. You could do quite a few figures just on this data. We're interested in soybean. So back to my soybean. We know that overall photosynthetic efficiency decreases, electron transport rates decrease, reaction centers are inactivated, and carbon fixation decreases. Okay, great. So why are you studying this, Jennifer? If we already know all this, why are you studying it? Well, all of the experiments so far, the cold exposure was 2 days or longer, right? So there's no information on really early. So they were all took place over 2 days, and mostly the temperatures were chilling, which is 10 to 15 degrees Celsius all the time, light and dark, or chilling during the day and cold only at night, right? So it's kind of up and down. And I just wanted to see what happened to photosynthesis if we were just like continually exposing it at cold temperatures for a short amount of time. So my questions are, what happens earlier, right? This is all 2 days or longer. What's happening in that first 2 days? Where is the damage occurring and when does that damage occur? Okay, yeah, the overall efficiency decreases. Why? What's driving that decrease? When? When does that decrease start? Is it different if my samples are light adapted or dark adapted? So those are the questions that we're going to be going through today. I did a short-term cold-induced photobiology experiment with 2-week-old soybean seedlings. That's them in the little picture. They were under 200 photons of light and all during the day and at 22 degrees C. And then at the beginning of the experiment, they were moved to 5 degrees. And I measured chlorophyll A fluorescence every 5 minutes for an hour, and then 15 minutes for an additional 2 hours, for a total of a 3-hour experiment, right? To like really get into this. All of this is in the light. So because I was doing every 5 minutes, that's not enough time for the photosystems to fully relax. So I did everything in the light. I used 9 plants per trial, and then my significance are based on ANOVA with Tukey HSD post hoc analysis. And this is literally how I would set them up. I'd take empty pipette boxes so that the clips wouldn't like pull the leaves down too far, and clip them. And then you can just open the door, pop on the Handy PEA on the receptor, go bam, 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 close the door. So you're not letting the cold out too much, right? Because that's going to change your temperature. So this is literally how I would set it up, is pipette boxes. It's very, very high tech. When we looked at this, yeah, soybean photosystem to its efficiency decreases in the cold, but it's really rapid. So in the light, like you start seeing the decrease after 15 minutes and it's significant at 25 minutes. So this is a really quick decrease in the efficiency. This is operational. So this is what the soybean is experiencing during the day in cold temperatures, right? Like, so here we go. And it goes down and it stays down for the rest of this small couple-hour experiment. The electron transport rate, which we talked about, also decreases rapidly. So this correlates with that linear electron flow through the Z-scheme that we looked at earlier, also begins to decrease at 15-minute and is significantly different at 25 minutes. So we're looking at things that are happening after 2 days, where the soybeans actually starting to respond to the cold photosynthetically in 15 minutes. Both the efficiency and the rate decrease similar. So of course, being me, I was like, okay, well, where in that linear flow is slowed down? So the efficiency and the rate are lower, but what's causing that? There's more than one spot in the Z-scheme where something could be going wrong. And that's where the OGIP curves come in from the Handy PEA. Remember I said it took every single time point between that FO and the FM? Yeah, this is what comes out of it. So you can see on the right side is just a normal OGIP curve that I made with just tester soybean data just to get my students used to working with it. And you have the O is the minimum, and then P is the maximum. That's why it's OGIP. So there are obviously 2 steps in between those. On the left, I have my diagram of the photosystem to reaction center. And one of the things that you have to understand when you're working with the Handy PEA and when you're working with Kautsky curves, which is another way of saying OGIP, is that each step is part of this reaction. So we have light, right? Light comes in and it hits the reaction center, and that excites an electron. That electron is then passed from the P680 up to QA, right? That first quinone acceptor. And that is the O to the J, right? So that slope. O to J, that's that moment in time. And then it's passed from QA to QB, and that's J to I. And then it's passed to the plastoquinone pool and beyond to photosystem 1, and that's P, right? So these actually correlate fairly well with these physiological steps. And I'm not going to go into all the math of that. There are some really amazing papers that will show you all of the mathematics that went into designing this. I can share them with you. They're on the PP Systems webpage. They are really great if you want the nitty gritty. Today, I'm just going to focus on the physiological. What does this actually mean? Not how did we figure it out? And then, of course, at the end, you have to replace that electron, and that comes from the oxygen evolving complex. So when we look at soybean in its cold state, you can see that all these OGIPs start to decrease at all points. So the very top curve in the kind of medium blue is 0 minutes, and then the navy blue is 15 minutes, dark gray is 25 minutes, light gray and light blue are 45 and 60 minutes respectively, right? So these curves are dun, 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 right? They're getting worse and worse and worse, and until about 45 minutes, and then they all look the same the rest of the way out, which is why I only went to 60 minutes on this chart. They all literally look the same for the rest of the time. So what you know that the electron transport is slowing because that's what it's measuring, right? It's measuring the electron going to QA, to QB, and then out through the plastoquinone pool. What I can see here when I look at this is that O to J slope is actually flattening, right? So if you look at that nice medium blue shade, it's a pretty steep slope to O to J. And then the slope starts to get flatter and flatter and flatter and flatter. And if you remember, the O to J was the electron moving from the reaction center to QA, right? So that is where the electron transfer is starting to slow down. The PI, the I to P section, kind of remains the same. Its slope and shape remains, even though it's lower in fluorescence, that shape is very similar. So it probably means that the plastoquinone pool through PS1 is relatively stable. So this to me suggests that soybean is getting messed up in that early electron transport state. The really cool thing, in addition to just looking at these curves, is all these different points, the O, J, the I, the P, and a bunch of the other points in between, can be used to calculate other parameters. So we can use these to calculate VJ, right, which is going to be the relative position of that J point, which is that first electron movement, to the min and the max. And then we have the slope, right? So that slope between O and J and how steep or flat it is. And then FV over FM, which we were talking about closer to the beginning as well, which is looking at the difference between the peak and the start. So I just put these in reference to the points on the curve. The equations can be a little more complicated, but this was more just so you could kind of get an idea of how it is being calculated by the Handy PEA. So again, remember that gobs of data, it just spits all this stuff out in a column. Like you don't have to actually do any of the math, but I like people to understand what's happening behind the scenes, not just, okay, yeah, great, data, woo. You know, what does it mean? What are these things actually telling me, and why are they important to measure? So photon trapping and photon dissipation are inversely related, which probably makes sense since we know that light goes one of 3 ways, right? It's either trapped and used, trapped and dissipated, or bounced back as fluorescence. And you can calculate these from the Handy PEA curve. So to look at trapping per leaf cross section is just taking FV over FM and then multiplying it by where it started. So that gives us how much of this light is being trapped. And then we can look at how much is being dissipated, right? Which is where we started minus how much is being trapped, right? So if it's not being trapped, it's being dissipated or it's being reflected back at us, which is what we're using to do these measurements. When we look at soybean, trapping decreases at 30 minutes and dissipation starts to increase really early, but it's significant at 20 minutes, which suggests that the soybean is trying to dissipate energy away from the reaction centers and from the photosynthesis because it's starting to get stressed out, right? So we've got 15 minutes, the overall efficiency starting to go down. Now we see dissipation is trying to send some of that energy off to take the pressure off the system. And then by 10 minutes later at 30 minutes, trapping is even going down as everything starts to fall apart. The active reaction centers, right? So the reaction centers that are capable of exciting that electron can also be calculated. And so this time you're using FV over FM and comparing it to the slope of O to J and that relative position of where J is between O and P. So this actually gives you the reaction centers that are active in that little circle that the Handy PEA is measuring. And in soybeans, the reaction centers are significantly lost at 25 minutes. And again, this is all in the light, so this is potentially reversible by allowing the system to be relaxed and not continually assaulted with light and cold temperature at the same time. And then the last parameter that we're going to look at, because there are at least a dozen more that I could have put in here, but the last parameter that is really common to use is the performance index. All right, so that's basically giving you a value of the overall efficiency and capacity of photosynthesis. So it's combining how many photons are absorbed, it's combining the efficiency of QA reduction, and it's combining the efficiency of the electron moving past QA. And that is its beautiful equation if you want to actually calculate it by hand, but the Handy PEA does it for me. So I trust its calculations way more than I trust myself in a calculator. The overall efficiency and capacity for soybean is significantly decreased at 105 minutes. So at 105 minutes, we have lost over both the electron reduction and the electron moving towards photosystem 1. So we're seeing an overall performance health. And this is the significant point. You can see on the curve that it's kind of a little up and down, and then it settles into being significant, right? So you have there's a little variety, and there always is, right? You never get perfect tiny error bar data. But this gives me an idea that after 105 minutes, soybean is really stressed out, right? The overall efficiency, the overall capacity of photosynthesis has significantly decreased. So let's look at this all together. What is going on in the light during cold stress in soybean? So after that first 15 minutes, we start to see a decrease in the overall efficiency. We start to see a decrease in electron transport. So let's, fade those out because we're starting to see it. And by 30 minutes, we have a decrease in active reaction centers. So there are fewer active reaction centers, a decrease in phototrapping, so less light is being trapped, and an increase in the amount of photons that are trapped being dissipated, right? So they're being sent back away. Now, there was no significant effect on the state of the PQ pool, which then leads to photosystem 1. But after 105 minutes, there was significant difference in the performance index, right? So overall, everything is being impacted by this cold. But this is under steady illumination, right? So they are in the light. This is only showing the working status. It's not showing the maximum capability. It tells us nothing on, well, can they recover from this, right? This is just what's happening when the light is on. So the next question is, well, can they recover from this? So I did this again, but this time I dark adapted my samples. And Unlike when they're in the light and some are open and some are closed because it's just doing its normal thing, I give it 10 minutes so that all those reaction centers settle down and are ready to go, right? So they're ready to accept the electron. And then you hit it with a burst of light. And so you get all of the ones that are active and capable being activated. And then that gives me the maximum. And this tells me if something really bad is happening because I let it relax. I let it get away from the stress, right? Photosynthesis is stressful for plants. The reaction centers break down and have to be replaced all the time. So you close it off and then you hit it. So we redid this. This is Allie. So that's Allie and all of her plants lined up so that she can just move the Handy PEA down the line. And same light, 200 micromoles, same light dark cycle. But this time we dark acclimated them using those clips for 10 minutes. So clips get closed. After 10 minutes, Allie places the receptor on it, opens the clip, hits the button, closes the clip, takes her thing back off. So we did this every 10 minutes for an hour, and then every 30 minutes for another hour and a half, and then every hour for another 240 minutes for a total of 420 minutes or 7 hours. Yeah, that was fun. 9 plants per trial, and again, I'm using ANOVA with Tukey post hoc analysis for my statistics. Now, when we are in the dark, right, so we give the photosystem time to relax between light pulses, you don't see a decrease in FV over FM until 360 minutes. And that photosynthetic index that I was just telling you about in the light, it doesn't significantly decrease at all over the entire 7 hours. So it's just, it's kind of a boring graph, which is why I didn't put it on here. So at this point, okay, yes, in the light, starting to drop at 15 minutes, but that is obviously reversible damage because when we repeat it in the dark, you don't, with dark adaptation, you don't see any difference in overall efficiency until 6 hours, right? 360 minutes. When you look at those OGIP curves and you look at the electron transport rate, these were not impacted during the 7 hours of cold at all. Right, so the I know the P on the 0 minutes looks a little taller, but if I had the error bars in there, it would not. It would just look because they overlap quite a bit. So there was no significant difference in the O, the J, the I, or the P at all during this experiment. The electron transport rate was not impacted by the cold after 7 hours, and the photon fate was not impacted by dark adapted cold. So you had trapping and dissipation. They just pretty much stay consistent throughout the entire experiment. But you do start losing some of those reaction centers. So, but it's 360 minutes or 6 hours before there's any significant loss of the reaction centers. And that is where you start thinking, okay, this is where some irreversible damage might be occurring because these are dark adapted. The loss of the reaction centers is becoming significant, and based on some other data that I have, it does keep going down and it stays really low throughout the next several days. When you look at this, it's decreasing at the same time as overall photosynthetic efficiency. So the photon trapping, the electron transport rate, all of that is the same, but we're starting to lose the number of reaction centers per cross section. So that is what's behind our FV over FM, right? Decrease. That's what's behind our efficiency. And that again is where I think the power from this particular instrument is. I'm not just getting the overall picture. I can start dissecting pieces of it out and being like, okay, well, what's driving this overall? And in this case, during cold stress, it appears to be the loss of reaction centers after 6 hours in the cold. So now we'll go and we'll think about the cold-induced photodamage in soybean. just in general, right? So those first 6 hours, we saw that there was a decrease in overall efficiency, electron transport rate. There was a decrease in trapping and increase in dissipation after 25 minutes in the light, but these were all reversible by dark acclimation. So these are not permanent damage. After 6 hours, the FV over FM decreases and there's a decrease in active reaction centers. And then after 2 days, because I have this data, but I didn't include it in the presentation, we can see that the electron transport rate decreases, photon trapping decreases, dissipation increases, and overall performance index decreases. So that 2-day mark that people have been using in the literature is a good spot, because that is where we start seeing irreversible damage, according to my research as well. So I did this for first 6 hours, and then I've also got every 12 hours as well, which I didn't get into. So 2 days does seem to be that breaking point for photosystem 2 in soybean. You also see that the PQ to photosystem 1 is also disrupted. So the whole thing is just starting to fall apart after 2 days. And it keeps going down, about stabilizes around that 5-day mark, which if you think back to the very beginning, I told you the LT50 is 5.2 days. And that 5 to 6 days is where the decrease in all these start to stabilize, and they're just really, really low. I'm hoping in the future with my students to examine non-photochemical quenching. So yes, they're being dissipated. Awesome. Where? Where are they going? So you can use light curves where you leave it in the light and you hit it with those pulses to understand the difference between 3 types of photochemical quenching, which I'm not going to get into right now, but there are 3 different types of photochemical quenching. I want to look at that reaction center stability because we know we're losing active ones. Are we losing the actual protein, or are the reaction centers themselves just not able to excite an electron? So to do that, that D1 protein where the oxygen evolving complex was attached in my diagrams, I'm going to look at it via Western blot. I have a student that's very excited to get started on that. She's never done protein blots before. And then I'd also like to look at chlorophyll content. So visibly, I can see that they're starting to get paler. And I'd like to be able to measure that, how much paler and when. That was one of those things that when you're doing the experiment, you're kind of noticing, man, you guys are getting more and more yellow, right? Your green is going away. But I wasn't set up to collect samples because that would kill the plant, and I needed it for the entire experiment to take my fluorescent samples from. So hopefully in the future, that's something else we can look at. Real quick before I hand it back to Carrie and take any questions. The funding for this work was provided through the IUPUI, which is my alma mater, their graduate assistantship, so during my PhD. The Niswinder Department of Biology at MU has a research grant that I was able to get that has been providing for my students. While I was at IUPUI, I had no money because I was a PhD student, and Tim Doyle generously mailed me his Handy PEA and let me play with it for like 6 weeks. So I really wouldn't have half this data without Tim, and he's here, so thank you, Tim, seriously. And with that, I am happy to take any questions that people have.
All right, Jen, well, thank you very much. That was an excellent presentation. A lot of good stuff there. And as Carrie mentioned, we're now open to questions from anyone. I would like to start, Jen, you mentioned a dark adaptation period of 10 minutes. Mm-hmm. Is that sort of a rule of thumb, or is it based on the low light that you were measuring or that you were growing in? Is there some sort of rule of thumb about the length of time needed to make sure you're fully dark adapted?
I actually tested it with several soybeans before I ever started my experiment. I had 2-week-old soybeans and I just started dark adapting for 5, 10, 15, 20, right? And so I did it experimentally based on my light conditions and to decide, okay, 10 minutes, I'm not getting a huge increase by 15, right? Like I don't see a big difference between my data, and I can get things done 5 minutes quicker. So that's where the 10 minutes came from. It's not a rule of thumb, and it does depend on what the external light is, what your species is, right? You can't, C3s are going to be different than C4, you know, like you can't just say, oh, 10 minutes. I've seen in the literature anywhere from 5 to 30 minutes, depending on light and all that. So I did it experimentally because I wanted to make sure that I was fully relaxing all of those before I ever started. So that was like literally the first 10 measurements that I took with that Handy PEA you mailed me.
Okay, great. Any other questions? So we have one sort of comment, Jen, that, great talk, but got lost with all the acronyms. So it would be helpful if you define them.
Yeah, the acronym soup gets really complicated. I have a one sheet cheat sheet that I actually print out for all my undergrads so that they can remember exactly what everything is. I went a little quick because I was getting excited. So, quick in talking with my hands is kind of what I do. And if you go back and kind of pause so that you can see, I tried to define the terms on the slide before, and then they're used on the next slide. And so if you just kind of, when the recording is ready, you can go back and kind of pick up on some of that. But yeah, I actually have a cheat sheet that I print out because it is alphabet soup.
Maybe that's, if you'd be willing to share that with me, I can sort of attach it.
Yeah, absolutely.
People, because that was a big interest for people, is just understanding all those crazy acronyms. So that would be super helpful.
Yeah, I made I call it my OGIP for noobs. It's a 3-page document that goes through the basics and then what everything physiologically means with no math that I give to my undergrads.
You can definitely see the instructional component to this. Like it was really well laid out. So I'm sure that's one of the reasons there aren't many questions, is it was just well done. So I think that, yeah, we had one person comment that no questions, but the presentation has been really interesting. Thank you.
Thank you.
So yeah, I would say, Jen, that we obviously really appreciate you taking the time to do your presentation. And we thank people for their questions. If anyone has any additional questions related to this material, feel free to email me, or reply to the email I sent out, or email me directly, and I'll be happy to get them to Dr. Robison to answer for you.
If you're on social media, you can also find me on Twitter and Instagram at Jen Robisi, because my last name's Robison and I do science.
Right. Okay, well, that was awesome. Thank you so much, Jen. And we'll get a recording out to everybody shortly. And awesome job. Thanks so much.
Thanks.
Yeah, thank you very much, Jen.
Thank you.
Thank you for attending this webinar. To learn more about the Handy PEA Plus, please visit ppsystems.com.